Can you close your door? I am Dr. Philip McMillan. Thank you for joining me. And today I'm going to be talking about an interesting piece of research that has come out of Japan. And essentially, they were looking at Omicron and trying to work out where it came from. So that's a very interesting point. And they've found some interesting news with regards to where it was first identified, slightly different from what we have been taught or told in terms of the mainstream media news. Critically, I think that it also highlights some important points that we have to reflect on with regards to how we deal with the next phase, I think, of this pandemic. And before I start, I'd just like to take the opportunity to remind everyone that coming up in just under a week is going to be the presentation that I've been doing, where I'm looking at the risk of infertility long term across the population. There is a link below for you to be able to register. What I call the infertility nightmare, and you can then join me next week as we discuss openly as to what I think is happening and what could happen in the future. So, back to the question with regards to Omicron and the origins of Omicron. So, this was first picked up in terms of Discovery News, and I had seen it. Somebody had shared this on LinkedIn. I thought this was very interesting. And so, this is the Discovery News on the 31st of August. They have said Omicron variants may have been artificially synthesized rather than naturally formed. And they are highlighting the two Japanese authors who investigated the evolutionary process that led to the formation of Omicron, and they were seeking to elucidate this evolutionary process, which had twice as many amino acid mutations in the spike protein as other variants. So I'm going to take you back again to basics. Just to remind you, this is a diagram of what the coronavirus looks like. On the surface, these blue areas here are the spike protein. The little red areas and orange areas, the tiny ones here, are M and envelope proteins as well. And inside, this is the cut section of the virus, you also have nuclear capsid proteins as well. These are all things that the body would tend to make antibodies against to be able to target the virus. And you can see here again the example of the spike protein. The spike protein is the entry receptor. It's like the key that opens the door to get into a cell. Okay? And that's the basics of how all viruses work. It just so happens that this virus, with its spike protein, tends to use ACE2 to enter the cell. So that's the basic bit. And so what they were researching was the variation in the spike protein between the Omicron variants and some of the other variants that had existed before. And just so you have an idea again, this is kind of like what it would look like. So this here was actually Delta. And the Delta variant, all of these red dots are the mutations that are different from the original virus. And then the Omicron, you can see they had significantly higher mutations. And that's part of the reason why it seems to be able to be getting past immune systems. So that's the basics that we're going from. And what the researchers were looking at, as they had pointed out in the Discovery News here, was they were trying to find where did it come from in terms of the evolution of the virus. So this particular paper was published on the 5th of August 2023. And you can see here, I have got, it's a preprint. That means that it has not yet been peer reviewed, but it was published on the 5th of August 2023. And you can see already it has 40,000 views and 20,000 people have downloaded this paper. And this is because people will want to look carefully at what they're trying to say. and make sure that it is accurate. But they are raising a very important point. What were the origins of Omicron? Now, I've said before that the fact that we have no clear answer to the origins of SARS-CoV-2, in my view, is an incredible failure of the scientific community. Because if you don't know that question, I'm not sure how you can understand anything else that goes beyond it. And I'll explain the clinical significance of that in a few minutes. You can see in the image here that I have Puerto Rico as number one. And in two, we have South Africa. So I'm going to take you back a little bit in terms of what was said originally when we had information coming out about Omicron. So I've got the paper here, the preprint. It's in a PDF format. And you can see here, almost a year later, these emergencies, the emergence of variants of concern, Omicron specifically, is a variant of SARS-CoV-2 first reported to the WHO by the Network of Genomic Surveillance in South Africa on the 24th of November 2021. This was when, sadly, because South Africa was doing the research of trying to find a variant, they were penalized. And suddenly, people couldn't fly from South Africa or Southern Africa to different parts of the world because they were trying to protect the rest of the world from this Omicron variant. It was all a little bit political. But it's interesting because when you look at the some important genomics paper, and this is another paper that I found here, and this paper actually was in August 2022, and they were looking specifically in Puerto Rico at SARS-CoV-2 and tracking the variants in Puerto Rico. They were doing this again from a scientific point of view to look at the evolution of the virus. What was interesting is that um, when they detected Omicron for the first time, it was detected on the 29th of November. That was their first case that they found. You remember, it was first detected in South Africa on the 24th of November. It was first detected, according to the paper, in Puerto Rico on the 29th of November 2021. So at that point, it seems as though South Africa is in first place, and they were penalized for this, both financially because of the politics, but also because they were doing the research. Here's what's interesting. When we go back to this paper that was done, so the Japanese paper was doing a really interesting bit of research, looking at all of the variants that had been identified in different parts of the world. And you can see here in the paper what they have found here. They were doing blast searches. That this is, I can't even remember what blast means, but it's a specific kind of search looking for sequences of amino acids, looking for the variants BA1 and BA2 Omicron variants. Look at this. We detected isolates of Omicron BA1 and BA2 lineages. Surprisingly, they found this. It was already present in Puerto Rico in 2020. Now, isn't that interesting? And they pointed out here that although South Africa was the 24th of November 2021, Puerto Rico had actually variants of Omicron in 2020. Really, really interesting. Why would that be so? You have to remember that when they completed this paper, and again, I'm telling you what the researchers have come to the conclusion on it. And you have to remember that in order to get it published, they have to be really careful with what they say. So here is what they have said at the end of the paper. They have said, we would like to add that while artificially synthesized viruses may have spread, they are not criticizing reverse genetics technology as it has made tremendous advances in virology. However, this paper aims to point out that SARS-CoV-2 has undergone unthinkable mutations under conventional coronavirus mutation mechanisms. And they hope the possibility of artificial theory should be included in the discussion seriously as to the formation of SARS-CoV-2 variants. So that's essentially what the researchers were saying. That's their conclusion. Essentially, what they said is that when they looked at this Omicron variant with all of these mutations, they just could not find how a coronavirus would be able to do this in that period of time naturally. That's technically what they're saying. So that's the science. And just to break it down a little bit more, I'll tell you why it's important with regards to Omicron versus the other viruses. I've shared this before, but it is such an important point. Most of the other variants before Omicron tended to primarily use this protein, TMPRSS, with ACE2 to enter very quickly into cells. And this is why, in theory, they would always outperform Omicron because they were therefore using the front door to get inside the cell. Omicron uses a much slower mechanism. It binds to ACE2, then it gets endocytosed, it goes in, then it's released. So this route is efficient but slower. So I had always asked, why would the virus switch to a slower and more efficient route unless this was blocked? And this is why I think that we had the Omicron, because as we said, it was identified in Puerto Rico in 2020. So it was floating around. Don't ask me who made it and why it was floating around in Puerto Rico. But the point being is that I suspect that the block of TMPRSS on the furin cleavage site probably led to the overgrowth of Omicron. And this is probably why it can't be controlled now. Because what's happening is that the primed human body with the spike protein is still targeting this entry mechanism and leaving this back door open. And I think that's my analysis as to why we're having significant spread of Omicron in especially highly vaccinated regions. It seems as though the immune system is not able to adequately recognize it and therefore stop it from spreading. So this is a real problem with regards to it. So people may ask, why, in any case, are you interested in this? I'm a clinician. I'm not a researcher per se. I'm not a virologist. I'm interested in it, and it's more curiosity rather than me dealing with and finding solutions to it. What I'm interested in is patients, because I want to know why patients are getting sick. I want to understand the mechanism because I have to see patients. I need to understand what's going on with them. And therefore, if we have an artificially created spike protein on a virus, remember that's technically what we're focused on, an artificially created spike protein, what was it designed to do? And this is why I think it is so important for us to get to source. We need to understand exactly what has been created, what was the purpose, who did it, and why. What was their intention? Was it designed to hurt? And if it was, in what ways? Because from a clinical point of view, if you don't fully understand the mechanism that is causing illness, you can't help a patient. And I think this is what we're seeing, especially with long COVID. We have this very strange situation across the world where we have two groups who are so far apart in terms of understanding what we're dealing with. One is very, very pro intervention, and the other one is completely against anything intervention and almost completely against anything like masking. I'm not interested in the politics, but I think both groups are missing the point. If we are dealing with an artificially created virus, we have a monster on our hands. And just to give you context, let's give you a little bit more context so you understand what I'm worried about. This here is a paper, and this was done in December 2022, and it's Human Interactions of SARS-CoV-2 Spike Protein, a systematic review. And they were looking at how many proteins interact with the spike protein. This is what I'm finding frightening. When we look at this, I'll have to find this in a second so that you can see what I was concerned about. I'm going to show you an image of here we go. I'm going to show you an image of how many proteins interact with the spike protein, and it's here. Now look at this. This is, and we can see here, this is showing us, let me just get this a bit smaller. These are all the tissues that it interacts with, primarily the lung, kidneys, other tissues, monkey tissues, and so on. That's one part of the problem. But even more concerning is the amount of proteins that it will interact with on a regular basis. So here we have, and I'll show you this one here. In this image here, what we're seeing here is how many proteins that the spike protein of the virus interacts with. ACE2, we know. Then there is TMPRSS, the whole family. Other miscellaneous proteins, the spike protein, furin. We have CD147, ADAM17. These are drifting down. NRP1. This is a really important one. And all of these proteins are interacting with the spike protein. Was that designed? And if it was designed, what was the intention in terms of causing harm? I'm sorry, I cannot accept that this is created for the fun of it. I call me looking at the glass half empty. But I just cannot imagine that if this was created in a lab, it's unlikely that it was created for the benefit of humankind. That's just my thought. I may be wrong. But even so, if it was, why doesn't the persons come forward and say why they were doing this for the benefit of humankind? And if they are not, there is a pretty good chance that if it was artificially created, as the paper seems to be suggesting, I remind you of what the paper said, the unnaturalness in the evolution process of SARS-CoV-2 variants and the possibility of deliberate natural selection. You can't get any more clear than that. That's effectively what these researchers are saying. As societies, as a world, at the end of the day, lives are either important or they are not important. Now, if they are important, we then need to understand what can hurt humankind, what can cause disease. And to put it in context, if the spike protein of the virus was designed, and this is one of the things that came out from Dr. Carlo Brogna in Italy, if it was designed to turn our microbiome against us, and this is what seems to be happening, where the spike protein seems to have bacteriophage properties, killing off good bacteria, strengthening bad bacteria, and helping them to make toxins. The outcomes of that is technically a slow poisoning across a population. How can we just let that happen? How can clinicians get patients better if they have no idea that that is happening? That's why answers need to be found, have to be found. And I'm sorry to say, but all those people who are listening, all those scientists, all those people who are interested in politics, if you do not have this as the top of your agenda over the next year or so to get answers and to find solutions, you are more of a hindrance than a help to society. That's just my thought. So I hope you found this information valuable, but don't just listen to it. Spread this information. Force people to come face to face with the possibility that there are people in the world who are so determined to hurt others that they will stop at no ends. Once we understand that, it gives us a chance to finding ways to mitigate a damage that has already started and for which we have no full understanding of the long term consequences. Have a good evening.